Uh, the online people can't hear at all, if anyone can hear us.
All right, can you hear us now?
Yes, thank you.
Awesome. No, thank you so much. I appreciate that. Alrighty, so now that we're all set, so people have been hunting wildlife for thousands of years, and meat from wild animals continues to be really integral to the food security and the cultural identity of hundreds of millions of people throughout the world. And this is particularly true for indigenous populations. However, the hunting, butchery, and consumption of wild meat is also a really important interface for zoonotic disease transmission. Zoonotic disease transmission from wild meat has been described in numerous studies, particularly in Central and West Africa and in Asia. For example, the emergence of HIV from SIV has been shown to originate at this interface, as well as others such as simian foamy virus, Ebola, and Marburg fleoviruses. I would not be a zoonotic disease researcher if I didn't say this statistic, which is 70% of all emerging and re-emerging infectious diseases are of zoonotic origin. An old mentor of mine used to say rats, bats, and monkeys, or primates, bats, and rodents are major sources of zoonoses. In Amazonia, however, zoonotic disease transmission from the hunting, butchery, and consumption of wildlife has been highly understudied. And this is really surprising, given that this region has been known to be a major zoonotic disease hotspot. Numerous pathogens have originated from this interface, such as Trypanosoma cruzi or Chagas disease, leptospirosis, and echinococcus. It is also estimated that about 1.5 to 2 million tons of wild meat is consumed annually in Amazonia. And so I'm going to start today's presentation here in this region of the world in Amazonia. This map here is taken by Han et al. In 2016, which predicts the distribution of zoonotic disease among mammalian hosts. Mammalian reservoirs of zoonotic diseases are globally distributed, but you can see here that there are hotspots notably seen in Amazonia. Further, indigenous reserves, where indigenous peoples own and manage their lands, is becoming increasingly common in Amazonia. These communities are highly dependent on wild meat for their food security. This potentially puts them at risk and puts them at the front lines of not only zoonotic disease risk, but of also being involved in surveilling of zoonotic outbreaks. And so partnerships between both scientists and indigenous groups is really necessary. All right, so what is needed? First is a better characterization of zoonotic disease risk in this region. And the second is to establish effective co-management programs with indigenous communities. First, to better understand the cultural beliefs and practices that may influence wildlife health and zoonotic disease, and then also provide long-term monitoring for wildlife health. In my research, I work with the Waiwai, an indigenous community located in an extremely remote region in the very far south of Guyana in South America. This is right at the northern border of Brazil. And I'll show a picture of where we work in the next slide. The Waiwai own and manage an indigenous reserve called the Kaneshan Amerindian Protected Area. The government recently changed the name, so I'm still trying to remember the new name. It is called the Kappa. And this Kappa is about 625,000 hectare reserve that is owned and managed by the Waiwai. There's approximately 300 Waiwai that are concentrated within the village of Masakanari in the Kappa. And The Waiwai are described, much like most of indigenous Amazonians, as forager horticulturalists. They practice a really traditional Amazonian subsistence strategy of swidden cassava horticulture, and then they supplement their diet with wild meat and fish. They are considered to be the most traditional indigenous group in Guyana, and hunting plays an integral role in Waiwai food security and also in their cultural identity. And this is particularly true for the hunting and consumption of primates. So here's a map of the Kappa. So again, ignore the legend that says Coca. It is now called the Kappa. The Kappa is outlined here in red. Essentially, no one lives sort of outside of the boundary of the Kappa. The Waiwai are concentrated in a village called Masakanari, which is starred here on the map. The Kappa protects one of the largest areas of contiguous rainforest in the entire world. And these large areas with really small population densities are really representative of most of the indigenous reserves throughout the tropical world. I thought it might be fun to show you some pictures of how we get to the Kappa. These are pictures from this past summer where we went by land. A colleague of mine kind of asked, why did it take you 2 weeks to get to your field site? And I think that these pictures answer that question. So it takes about a week to go by land, and then once we hit the river, it takes about a week to travel by the river in a canoe. And here is a video, if I can flip, of traveling by river in a remote region in the Amazon rainforest going to Masakanari. I'm going to transition now to talk about how the Kaneshan Ecosystem Health Project was established. In 2013, at the request of the Waiwai, Dr. Christopher Schaefer, who is an environmental anthropologist at Grand Valley State University in Michigan, started working with the Waiwai. And this was to address questions regarding sustainable resource use and understand the sociocultural importance of hunting and fishing. In 2015, I started working with the Waiwai to develop a health component of the project that initially focused on assessing and mitigating zoonotic disease risk from wild meat consumption. From these collaborations, Chris Schaefer, myself, and the Waiwai started the Kaneshan Ecosystem Health Project, which is an indigenous scientific collaboration for the co-management of natural resources and the promotion of shared human-animal health. Chris and I have been going to the Kappa every year, living and working with the Waiwai since the inception of this project. And shameless plug, Chris will be here for the next conservation science seminar where you'll be able to hear about his work. As I alluded to in my introduction, much of my wildlife disease research is really informed by both qualitative and quantitative immersive methods, particularly from ethnography, which is taken from anthropology. Ethnographic approaches both systematically and inductively characterize the sociocultural context. And they really do remain highly underutilized in both wildlife health and in zoonotic disease research. One of the first questions that developed from our ethnographic research working with the Waiwai and that was identified as a community priority was this question: are primates in the Kappa healthy? This question is a community priority as primates, again, are vital to both Waiwai food security and cultural identity. So I think this question really serves as the starting point of our community-based wildlife health program in the Kappa. And this program has been building since its inception in 2015. And I think what's really special about this co-management program is that we really do sit together to develop our research questions. One, that these questions are addressing community needs and priorities, and they're also addressing important questions for the broader scientific community and the creation of new knowledge. And there's a real recognition from both parties on the needs and interests of both parties involved. And I think that this really demonstrates the trust and the honest conversations needed to do this work. And I think that you can really only get here if you are consistently going to a community over a long period of time. So I'm going to use this timeline throughout the remainder of this talk to show how our research and research directions kind of lead into one another and demonstrate the cohesiveness of this research program. So I'm going to dive now into some of our long-term and ongoing research projects in the CAPA. Before I get started, I do want to acknowledge all of the members of our team, including both the YY and scientific collaborators. Many of you all are in this room. I was planning on putting your pictures up during various projects that you're involved in, but it got too unwieldy, so. I want to start with our ongoing hunter-based disease surveillance program, again, that was started in 2015 to address this question of whether or not primates in the CAPA were healthy. These are some pictures from our initial training program where I trained 5 YY hunters on how to conduct a field necropsy on primates. So here I'm training Elijah on conducting and collecting tissue samples from a monkey he just harvested. In the upper corner, we're having a workshop where we're developing culturally appropriate methods for long-term data collection. So even as we speak, even as we're sitting in this room, the YY are collecting tissue samples. From 2015 to 2019, we conducted 127 primate necropsies, and the vast majority of these necropsies were conducted by YY hunters themselves. For general primate host health screening, we submitted 82 primates, which totaled 406 tissue samples, including samples from the heart, lung, liver, spleen, and kidney. And we submitted those samples to the University of Illinois Zoological Pathology Program to be reviewed by our collaborator, Dr. Karen Terrio, who is an exotic and zoo animal pathologist. Dr. Terrio and her team then read the samples or read the tissues and wrote up necropsy reports for each of the primates that detailed both the morphological descriptions of those tissue sections and provided a morphological diagnosis. We then analyzed the data for disease patterns among the sections. Now, I'm not going to get into the details of the histologic sections. Maybe some of our vets over here could at a later date. But the major take-home message here is that the primates in the CAPA are healthy, that there's really no significant patterns of disease among these sections. I think this work really sets the stage for some really important research moving forward. First, the community is on board. Since 2017 now, hunters have been collecting tissue samples, not just from primates, but they were interested in building this program to collect tissues from all wildlife species harvested for food. This program has really given us this unparalleled cross-sectional sample of wildlife, and the continued sample collection will allow us to continually monitor wildlife health populations into the future. In addition, in 2019, we started collecting molecular grade samples for pathogen surveillance and have already started to train YY hunters in taking those samples. The community is also highly interested and invested in building a field-based laboratory. And we tested out this field-based lab this past summer, much to the credit of Lexi Frank and Dr. Evan Kipp. And this includes parasitological and molecular equipment. And there are two main reasons why I'm really excited about this approach. One, yes, we can answer really cool, molecularly focused questions in the field, in the middle of nowhere, in an area that is highly understudied. But we can also close the loop. And what I mean by this is that we can have everything from data collection to laboratory work to analysis done in the Kappa alongside the people that we work with. And because of our long-term partnership, we can do this year after year, building the next generation of Indigenous and scientific partnerships and scholars. While conducting field necropsies and ethnographic data, I also noticed that no matter how the YY discarded the entrails of the animals that they were butchering, that YY dogs would always find them and always consume them. Dogs are really at the center of the human-wildlife interface in the Kappa. So this really got us interested in how dogs could serve as intermediaries or bridges of disease transmission between wildlife and humans. Among many indigenous Amazonian groups, this interface between humans and dogs is particularly pronounced. Dogs are often used in hunting. They spend a lot of time in the forest, which increases their opportunities to interact with wildlife. And dogs are extremely important culturally and economically. Dogs have been implicated, however, in this region to transmit a variety of zoonotic pathogens, such as leishmaniasis, Chagas disease, and echinococcus. The YY in particular have been known throughout the Guyana Shield for having the best dogs. The YY have traded their dogs with other indigenous groups in the region, and they still do so today. Traditionally, YY dogs are really well taken care of. So it's kind of hard to see, but the dog pictured at the bottom is wearing a feathered collar, and the dog right here is being breastfed. Cultural practices related to dogs and hunting prowess still remain today. Here, Elijah is in the middle of butchering a tapir, and he's rubbing this young hunting dog with tapir blood. And this is believed to make a dog a better hunter. In 2019, we set out to research whether dogs could again serve as these intermediaries or bridges for disease transmission between wildlife and humans. We did this through conducting physical examinations, collecting biological samples, including blood, as well as conducting structured interviews. And in one of our interviews, we asked, has your dog ever been bitten by a vampire bat in the past year? And 70% of respondents said yes, they had. This vampire bat dog interface is also consistently mentioned while we were conducting participant observation and unstructured interviews. We have also observed vampire bat bites on the tip of dogs' ears and nape of neck while we were conducting physical exams. However, when we probe more about rabies and the potential clinical signs, no one in the community had any sort of understanding of what that was, nor had they ever seen behaviors associated with rabies. This interface is a research direction, this rabies vampire bat interface is a research direction that we're currently pursuing. As part of her dissertation research in the College of Veterinary Medicine, Lexi Frank, who is there and is also pictured here, led bat research this summer in the Kappa, specifically targeting vampire bats. A lot of indigenous communities throughout the Amazon have vampire bat populations living with them and experience rabies outbreaks. And we're interested in exploring the question of why vampire bats are not a problem with the YY, but we do see issues of vampire bat and rabies outbreaks in other indigenous communities, particularly in Guyana. Another question is also, how do changes in local ecological knowledge and subsistence strategies lead to the emergence of zoonotic diseases such as rabies? Or how can these changes buffer or prevent against zoonotic emergence? And to explore these questions, we're really interested in pursuing future data collection efforts where we are looking to ID bat roosts, figure out where these vampire bats are, where are they going, who are they feeding on, and how frequently do they feed on dogs. Another major community priority that has been identified through our ethnographic data was this concern over mosquito-borne diseases. The YY Village Council asked if we could start to do some research to understand arboviral risk in the Kappa. This request was really prompted by observations from community members who had returned home, who were participating in mining camps outside of the Kappa, and they were experiencing symptoms that were really characteristic of mosquito-borne diseases, such as painful and swollen joints, fever, and malaise. But the YY do not perceive of mosquito-borne diseases as occurring within the Kappa boundaries, but they associate mosquito-borne diseases in areas outside of the Kappa where there's a large amount of anthropogenic disturbance, such as in these mining camps or in large-scale agricultural areas. This project is an absolutely huge team effort, and I want to acknowledge both the field and academic contributions of both Chris Schaefer, Philip and Stephen Sousay, Elliot Oakley from the Field Museum, Evan Kipp, Tiffany Wolf, Kim Vanderwall from the vet school, and Adam Hendy, who is now the adjunct assistant professor here in FWCB. The observations that the YY were making about only perceiving mosquito-borne diseases in these anthropogenically disturbed areas really does make sense. The emergence and reemergence of infectious pathogens have been linked to human-induced land conversion and associated habitat fragmentation. This is especially true for mosquito-borne pathogens such as yellow fever virus. Yellow fever is really interesting. It is maintained in the selvatic or forest transmission cycle that essentially involves canopy-dwelling mosquitoes and non-human primate reservoirs. Yellow fever, though, is really extremely vulnerable to landscape changes. So alterations in these landscapes can influence the abundance as well as the distribution of these yellow fever mosquito vectors, as well as their vertebrate reservoirs. So when we see landscape changes, oftentimes these mosquito vectors that are carrying yellow fever will go down to the ground and change the vertebrate reservoirs that they're feeding on. In South America, yellow fever is the most clinically severe arbovirus, and all human cases occur from the spillover from the selvatic or forest system. Interestingly, despite knowing about yellow fever for a really long time, having a really effective vaccine, we really know little about the selvatic cycle of yellow fever. Yellow fever, what we know about yellow fever and the selvatic cycle of it is really coming from data taken from highly fragmented forests, which really do not capture selvatic transmission dynamics. Additionally, we know that non-human primate demographics and ecology really constrains yellow fever spillover, but that too has been really understudied in selvatic forest environments. So the main objective for this study was to understand how endemic yellow fever virus is maintained in intact forest environments and how gradients of anthropogenic disturbance perturb normal sylvatic transmission. We received an internal grant from the College of Veterinary Medicine to do some initial work this past summer. We also submitted an NSF EEID grant this past November. I am unclear whether or not that grant will be reviewed. The objectives of our NSF grant was one, to determine the community structure and seroprevalence of yellow fever virus mammalian hosts across a disturbance gradient. Second, to assess how disturbance variables influence yellow fever vector ecology. To utilize phylogenetic analysis to understand multi-host transmission dynamics, and the environmental factors that influence yellow fever dispersal. And then lastly, to develop a mechanistic model to address fundamental questions about the conditions required to sustain and the mechanisms underlying emergence of yellow fever in sylvatic environments. For the purposes of today's talk, I'm just going to focus on some of the preliminary data that we did this past summer, assessing how disturbance variables influence yellow fever vector ecology. To do this, we constructed two 15-meter high platforms in the canopy. One platform was constructed in a highly remote, undisturbed location in the Kappa, and then the second 15-meter platform was constructed in an old fallow farm, which we described as our disturb site. We then collected mosquitoes targeting yellow fever vectors for 6 days at each site. Essentially, one person was on the platform with a butterfly net, collecting mosquitoes one by one and sticking them in a vial. And then there was another person on the ground level doing the exact same. We then morphologically identified the mosquitoes using a stereo microscope. And by we, I mean Evan Kipp did all of that work. And then for just proof of concept, we molecularly identified mosquitoes just to confirm the morphological identification. And I thought it also would be fun to play some videos. This is a video of us constructing this 15-meter platform, really in the middle of one of the most remote places in the world. And so Philip and Stephen are constructing the platform, and we're bringing large logs to bring up to them. The YY have historically built platforms up in the canopy to catch birds, and so this was something that they had known how to do and how to do really efficiently. And then this is a video that Evan took on the platform in the undisturbed site. And I just think that the image is just amazing what you can see so high up and 15 meters in the canopy. During this period, we collected over 1,000 mosquitoes in less than 2 weeks, both at the undisturbed site and the disturbed site. And again, we're targeting these yellow fever vector mosquitoes, particularly from the genera Haemogagus and Sabethes. And as you can see from this table, the percent of Haemogagus and Sabethes mosquitoes relative to the total number of mosquitoes that we collected were essentially equal at both of the sites. However, we do see some interesting differences in specific species of these mosquitoes. So Haemogagus janthinomes is the primary vector for sylvatic yellow fever. In the undisturbed site, we found Haemogagus janthinomes was highly abundant, particularly in that canopy. And that makes sense given that it's a sylvatic yellow fever vector and it's probably hanging out in the canopy to feed on non-human primates. In contrast, in the disturbed site, this species was almost entirely absent from any of our collections. Interestingly, too, we found that more non-target mosquitoes at the disturbed site, both in the canopy and on the ground. But at the undisturbed site, there was really no non-targeted mosquitoes in the canopy, but we did find them fairly abundant on the ground. All right, I'm going to kind of switch over now to talk a bit about mercury contamination in subsistence fish. Mercury contamination is one of the greatest threats to conservation and human animal health in the Kappa and really within Amazonia more broadly. There has been a recent development of a small-scale gold mine within the Kappa boundaries. And inland fisheries are the main source of protein for the YY, and any alterations in this food supply or chemical contamination can really expose the community to really unprecedented levels of food insecurity. So we started a project to study mercury contamination in subsistence fish in the Kappa. Amazonian forests are considered to be ecosystem sinks for atmospheric mercury. Again, one of the most dangerous contaminants to environmental and human health. Fish consumption is the primary way in which humans are exposed to mercury. And again, this is really concerning for indigenous Amazonians who are highly reliant on fish for their subsistence. And as these communities begin to be more and more incorporated into the cash economy and have more reliance on the cash economy, many indigenous communities now, like the YY, are engaging in these resource extractive activities such as gold mining. And with gold mining, essentially, they use mercury to amalgamate gold particles. Artisanal and small scale gold mining is the primary source of anthropogenically induced mercury emissions in the world. It's estimated that about 30% of global emissions are connected to gold mining directly in the Amazon. In 2018, the Kappa received this protected area status from the Guyanese government. But in 2020, some community members started a small-scale gold mine in the Kappa. And this activity remains really contentious, both between the YY and the Guyanese government and the YY themselves. The Guyanese government has threatened to revoke the YY's protected area status if the mining continues on their land. So in 2022, the Tushao, or the YY chief, approached us and collaborators at the Field Museum to start to study the effects of gold mining in the Kappa. And there were also a really growing number of concerns from community members about the negative impact that this mining operation would have on subsistence fish and human health. We recently received a small grant to do pilot work, and this grant was from the Institute on the Environment to study mercury contamination in fish and fish health in the Kappa. The first objective here is led by Chris Schaefer and Elliot Oakley from the Field Museum to employ ethnographic methods to characterize YY communities' perceptions of gold mining and associated impacts on fisheries resources in relation to traditional forms of anthropogenic land use change. The second aim is the aim that I'm leading to understand how mercury burden on subsistence fish consumed by the YY in relation to small-scale gold mining. So I'll focus on some of the research that we did over the summer. This past summer, we conducted some pilot data for this project. We conducted necropsies on 14 subsistence fish collected at various distances upstream and downstream from the mine. We actually conducted more than 14 necropsies, but a mysterious rat came in at the middle of the night and stole a bunch of our muscle samples. So we collected a piece of muscle from the fish, and then we put it in a food dehydrator to desiccate that muscle in order for it to be submitted for mercury and methyl mercury screening. We also collected organs for histopathology, again, to look at overall fish health. And then we collected molecular samples for species identification. In addition to this new research direction, I'm also really excited because this also represents a new team working together. We are working with the USGS Mercury Research Lab for mercury and methyl mercury screening. We're working with Ryan Lepak from the Environmental Protection Agency located in Duluth, as he is an expert on mercury screening in subsistence fish. And then this research has also been identified as a major priority for our collaborators at the Field Museum, and we're starting to work together to figure out how we can pull our resources to apply for larger funding for this work. I also have the opportunity to work with Nick Phelps, who has been helping give expertise on aquatic ecosystem health and ecotoxicology. Alrighty, so I'm going to transition now to talk a bit about some of my current and future research projects working with the White Earth Nation. And I see some really exciting parallels in both methods and in future research questions between the work that I just described in Guyana and the work that I've been doing in White Earth for the past year. For the past year now, I have been working in White Earth to assess the needs and potential opportunities to establish a hunter-based chronic wasting disease surveillance and management program. For this research, I have been spending a significant amount of time living and working in the community and conducting in-depth interviews with hunters. I want to pause for a moment and acknowledge my collaborators on this project, particularly Doug MacArthur and James Uran Jr., for which none of this research would be possible without their leadership and contributions. I also want to acknowledge Tiffany Wolf, who is the PI on this project, as well as David Fulton from FWCB and Keith Carlisle from the USDA. As many of you know, chronic wasting disease, or CWD, is a disease that is affecting white-tailed deer populations in Minnesota. And there's a massive effort by both state and tribal nations to increase the surveillance of chronic wasting disease through the submission of deer lymph node samples that are harvested by hunters. And despite efforts and incentives to submit samples, participation by hunters has remained somewhat low. Additionally, tribal communities such as White Earth have identified a subset of hunters who either I self-identify or other community members identify as community hunters. These are hunters that hunt for individuals that cannot hunt for themselves, so either for elders or for ceremony. Understanding the perceptions of chronic wasting disease from community hunters is really vital for chronic wasting disease management and surveillance. Since January of 2024, I have been working with Jimmy, who's in that black sweater here, on conducting these really in-depth, unstructured interviews among community hunters and among people that are really reliant on venison for protein. These interviews often last hours long, and they are really open-ended. So we talk about perceptions of chronic wasting disease, attitudes on testing and surveillance, barriers and opportunities for hunters to participate or lead in management for chronic wasting disease. We also have been going to a lot of community events where we have been conducting this ethnographic mapping project, which I'll talk about in a few slides. Preliminary data from this work show that participation in chronic wasting disease surveillance is limited due to concerns by community hunters about tag limitations. So in White Earth, hunters only receive one tag for the deer season. And so if they're hunting for other individuals or hunting for ceremony, they're often taking more than one deer. And they're concerned that if they drop off 10 heads at the White Earth Department of Natural Resources, that they could get in trouble for overharvesting. This is despite the fact that the Department of Natural Resources and White Earth have tried to really communicate that they do not need a deer tag in order to test their samples for chronic wasting disease. So some of the quotes that kind of support this, someone said, ain't nobody going to tell me that I can't, or stop me from hunting and feeding my people, and no one is going to tell me what I can or cannot do on my own land. Another theme that arose from our interviews was that less and less people know how to appropriately butcher a deer. If they do hunt, they often may not utilize the entire deer carcass. And this can be a really important, especially in regards to chronic wasting disease, could potentially increase the risk of chronic wasting disease on the landscape if that animal remains on the land. There's been a real push by community hunters and saying that there's a real need in the community to increase the education on how to appropriately butcher a deer. So there are some quotes from this. It's in the family. There's probably a fair amount of people who don't know what to do. White Earth has to see themselves in it, meaning they have to see themselves in understanding how they can contribute to helping with CWD in order to do something about it. In addition to the in-depth interviews, we also conducted a pilot ethnographic mapping project. Ethnographic mapping is used to understand the environment where activities take place from the participant's perspective. And this really helps to connect people with place, to identify key activity or places of key activities, to identify locations and boundaries that have meaning and value from the participant's perspective. It can also help to plan interventions and to recruit participants. And that's exactly what we did for this portion of the project. When we went to community events, it was a really great way and a great way to serve as an introduction to get people to talk about hunting and to want to get them to be interested in doing interviews with us. So we would ask white earth hunters at community events to put various pins on this map. And so we would ask them, place a blue or a green pin in a location where you last went hunting. And again, this really allowed for people to start talking about hunting because people really like to talk about hunting. And then we'd ask them, put a yellow pin on the map where you think a good spot would be for a deer head drop off site. So people drop off deer heads in order to test for chronic wasting disease. And then we'd also ask, put a red pin on the map where you think a good spot would be on reservation boundaries for a carcass disposal site. I'm really excited to continue to use more ethnographic mapping approaches. We'll be working with Chris Schaefer on building a GIS that links both cultural knowledge to deer hunting and CWD surveillance and management in the future. Now that we've completed our interviews, we are currently identifying community hunters in White Earth to participate in a hunter monitoring program. And we're hoping to have this up and running for next deer season. And we're also currently in the process of working with White Earth Natural Resources to kind of figure out the kinks regarding tag limitations. We recently had a hunter training workshop to begin to teach community members how to extract lymph nodes from deer to test for chronic wasting disease. And as you can see, this work is really similar to the work that I've done in Guyana, building a hunter-based disease surveillance program. Again, the hunters are extremely knowledgeable about the animals that they hunt and can easily get the tissues required for disease surveillance. Additionally, Dr. Tiffany Wolf and I received a USDA grant to develop animated and social media videos specifically targeting tribal communities. So these videos will be made by and for tribal communities. These videos will focus on butchery practices, lymph node removal, and just basics on chronic wasting disease. So again, I see really major parallels between the work that I do in White Earth and the work in Guyana. And in establishing future work on White Earth, I have really gone about it really similarly to how I approach the work establishing the Kaneshan Ecosystem Health Project. And that is, again, through ethnographic data collection and community-driven priorities is what will drive these research directions. One major thing that came out of these in-depth interviews was that yes, CWD is a concern within the community and among community hunters, but also is access to venison, both culturally and for food security, and that is a more of a pressing issue. And so I'm really interested in expanding upon our initial research studying chronic wasting disease to a more holistic approach studying subsistence hunting and protein sovereignty. particularly interested in studying how cultural norms and maybe food taboos may dictate the abundance of animal populations on the reservation. And because I see so many parallels between these communities and the research that we're doing, in October of 2025, Philip Sousay, who is the YY Kappa manager, will be coming to the Field Museum, to the University of Minnesota, and will also come to White Earth, and we'll do a bit of a cultural exchange. And we're hoping that this really sets the stage then for someone like Jimmy Uran to come down to the Kappa with us. So I'll end with just a few concluding thoughts and future directions about my future lab in FWCB. I think that these two research programs, both the Kaneshan Ecosystem Health Project and the work that we're establishing in White Earth studying protein sovereignty and wildlife health, can really serve as pillars for a biodiversity and health lab in the Department of Fisheries, Wildlife, and Conservation Biology. Given the global threat of emerging infectious diseases, and that many Indigenous communities are really at the front lines of zoonotic disease surveillance and management, it's really imperative that scientists work collaboratively with Indigenous populations to understand zoonotic disease emergence. There is a real need for long-term partnerships and the use of mixed methodologies to provide that contextual data needed to understand wildlife transmission risk pathways. And I hope that the Biodiversity and Health Lab at the University of Minnesota in the Department of Fisheries, Wildlife, and Conservation Biology can really serve as a blueprint or a model for these Indigenous scientific-led partnerships. Want to just take a few moments to thank all of the people that I work with. I'm extremely lucky with the team that I work with. They're wonderful. So thank you all for the folks from the Kappa Ecosystem Health Project. Would also like to thank funding and institutional support. And then thank the folks from the White Earth Protein Sovereignty and Wildlife Health Project. And Dr. Roger Faust, a graduate of the program, will also be working together on this project. And then also like to thank our funding and institutional support. And with that, I'm happy to take any questions. Alex.
I got hung up on something as you were talking.
Oh boy, okay.
And it's a me problem, not a you problem, but I think it might speak to something you probably deal with all the time in your research. So I was thinking, words don't mean the same thing between languages, and they probably don't mean the same thing to speakers of the same language. So I was thinking, healthy. That can mean hale and hearty, that can mean nutritious, that can mean safe, that can mean happy. And maybe it means all those things to some English speakers and almost none of those things to some others.
Yeah.
How, when you're working with one of these cultures, do you approach that challenge of like, how do we know we're even talking about the same thing? Try to understand what their interests and needs and concerns are.
That is a wonderful question, right? Yes. Okay. All right. So how do we what is the definition of health, right? And between all of us, we probably all have very different definitions of what healthy is in this room. And there are also various definitions of what healthy is between different cultures too, and between different speakers, right, of different languages. And so I think that's kind of where this anthropological approach is really important. So when you live and work with a community for a long period of time, you get this, it's called like the emic perspective, which is the insider's perspective. And then in anthropology too, it's really important to have the edic perspective or the outsider's perspective. And you use both of these perspectives when you apply it to your research. And so when we have the emic perspective, when we're within community, we can start to see kind of how they perceive of health. And also just talking to people too, right? So much of the work is just talking through what it is that we're doing and what it is that we're doing even before we do it. So that there is kind of this like baseline understanding that we're kind of on the same page. So we never actually like go into a community and it's just like, all right, let's collect samples now. Like it takes years and years just to even start to collect those samples. And I think that kind of goes to your question of, I think it really takes time to sort of come to an agreement and understanding of what we think health is and what we're looking for. And then also, you know, it doesn't always have to necessarily align. So there's a project, Rob, cover your ears, where where there was a parasite living in spider monkeys in the abdominal cavity. And to me, when I initially saw it, I was like totally freaked out. I thought, you know, this was a big problem. The YY were like, oh no, that's nothing. It's no big deal. And so we did a bunch of research to understand that, both through molecular methods, histopathology, and anthropology. And we found that this was a non-zoonotic, non-pathogenic parasite of monkeys. And again, this is, you know, for me, this animal was not healthy because it had a parasite, but the why we were like, oh no, it is healthy. Don't worry about this. And so we were able to kind of like align both of these knowledges to actually confirm that yes, the why we were totally right. It is just completely harmless parasite of an animal. Yes.
Stated that their concern was our wildlife healthy. How much of that is for us to eat versus for the sake of them as a prey for populations?
For the YY, it's about them eating, maintaining it's actually, I mean, it's both, right? If they're not healthy, populations are going to collapse and they're not going to have any food to eat. But for the question in this regard, it was very much about, do we have enough primate populations to eat going forward? So the YY had been traveling to other indigenous communities in the region who also consume non-human primates. And those communities were telling the YY that their primate populations were plummeting. And that they were also hearing that primates are not safe to eat. And so the YY were really concerned about that. And so that is one of the reasons why they reached out to Chris Schaefer to see if he could come in and start modeling primate populations into the future. And so that's actually something that he'll be talking about next week, is how he does that. Yes, Rob.
I want to thank you so much for not showing us monkeys.
You're I know. I'm sorry that I
This is a really simple question. I noticed at the beginning of your talk, you never used the term bushmeat.
Hmm.
Is that a term that's not appropriate anymore? Or
So language changes over time, you know, and it cycles through. And so right now there's kind of like a taboo against using bushmeat,
okay,
but honestly, the YY use the term bushmeat. So when I'm when I'm working with the YY, we use the term bushmeat. And so, you know, language matters depending on what context it's used, kind of what cycle it's going through now. So I'm using wild meat because it's what is accepted now. May I be using bushmeat in a few years? Yeah, I mean, it cycles in and out. But there is kind of this, and it makes sense too, when we talk about eating wildlife, we don't say that white-tailed deer and venison is bushmeat. We use bushmeat when it relates to people living in the tropics, hunting animals that we may perceive as not appropriate sources of food. And so there is that like negative connotation and location connotation. So I think wild meat works because it kind of applies to every.
changing my ethics lecture.
Yeah.
Yeah, first of all, great talk, it's really interesting, but my question is basically, it's pretty broad, but it's about culture shock. So in the initial parts of working with wildlife, what culture shock did you have? And how has that progressed over the years while you continue to work with them?
Yeah, I think culture shock is extremely important to talk about, to acknowledge. It is a thing. And if people don't acknowledge it, I actually think it's a huge problem in doing field-based research, is not accepting culture shock, not allowing people once they come back home to take time to kind of readjust. So when working with the YY, yes, there's a big culture shock because you're in such a remote region and you think, like, I just assumed I would have such an immense amount of culture shock coming home. But I actually, for whatever reason, don't have as much as opposed to some of the other areas of the world that I've worked in. And I think a part of it is because I'm living in a village and I'm working really closely with other people. So I think that like human connection is really there. And even now, like I have YY family, so it's a different feeling. But you're really remote, you're really separated from kind of the outside world. But areas of the world where I've worked in previously, where I've been working in a remote region, but not in communities, that's when I really felt culture shock. And that it's just like getting back into like normal socializing was really strange. But you could probably also ask Lexi and Evan, who came in to the Kappa last summer, kind of how they experienced it, because that was their first time.
Okay, well, thank you, Marissa, and I'm sure she'll be glad to answer additional questions if you want afterwards. And could the Prime Science students drop over and put your initials on the sheet? Thank you.